Hello, my name is Kamran Mirza, and this lecture is on acute inflammation. The material in this lecture is mainly from Robbins and Gotran, pathologic basis of disease. And so today, in two lectures, we will be covering acute and chronic inflammation. So, when we think about acute inflammation, these are the features of acute inflammation, and this is something that has been known for a long time. So, even Marcus Aurelius and all of these people before, Aurelius Celsus and the Virchow also was talking about what acute inflammation is. So, think about it for yourselves. When there's an acute inflammation, typically it's because of injury. That's the first thing. Then there is going to be rubor or redness, right? Calor or heat. There's going to be growth in the size, which is tumor. There will be pain, which is dolor. And then there will be loss of function of that area as well, right? And so these are the cardinal features of acute inflammation. And these are what these from time immemorial we have been looking at, right? And so if you have a bug bite or if you have a cat scratch, all of these are the features of acute inflammation. So acute inflammation, therefore, has there been studied by multiple people. And so here are some names of the individuals that have been looking at acute inflammation. But basically, what they found is that once you have an injury, there is a lot of leukocyte migration to that site. So here you can see this is the injury. This could be a bug bite, for example, or like a splinter. And you can see that the blood vessel close to it is responding to it. And the way it's responding is by sending its leukocytes because of the bacteria that has come in. And this bacteria causes an acute inflammatory response, which will cause injury, rubor, calor, tumor, dolor, and loss of function. Now, lots of things have been studied about acute inflammation. There's a play about, people with acute inflammation. There is a huge pharmaceutical industry talking about acute inflammation with all of the medicines that are anti-inflammatory. And there's a lot of research happening about acute inflammation as well. And so, if you want to define it, you have to define acute inflammation as an inappropriate inflammatory response with no foreign substances to remove. It's the basis of autoimmune disease, right? So, autoimmune inflammation is obviously the inflammation that goes wrong, and it's typically because of an inappropriate inflammatory response, and there's no foreign substance that we want to remove. So, inflammatory processes must be tightly regulated by the immune system to avoid excessive tissue damage and spill over to normal tissue. So, what happens is that inflammation can cause a lot of damage because normally inflammation will be done so that it removes an offending agent, and this could be a foreign body, it could be bacteria, it could be viruses. But when there's nothing to remove, that's autoimmune inflammatory response, and inflammation has to be tightly regulated. So let's define it. Acute inflammation is the reaction of vascularized tissue to injury. So think about this, vascularized. That means that the tissue has to have blood vessels. If there are no blood vessels, you cannot have acute inflammation. So for example, in the nail and in the hair, there's no blood vessels here. So you cannot have inflammation of the hair. You cannot have inflammation of the nail. You can have inflammation of the nail bed beneath the nail because that's where blood vessels are. Typically, you cannot have inflammation in the iris as well because there's typically no blood supply. But if you have iritis, it's when the blood vessels will come in, and then in cornea and etc., you will have inflammation there. So the culmination of this tightly regulated complex series of interactions is occurring will be acute inflammation, and the interactions are between the pathogen or the injury, the host inflammatory cells, the complement cascade and the coagulation cascade, and also the release of chemokines and cytokines, which are inflammatory mediators that will activate the leukocytes. And what's the end game? The goal is that you want to neutralize the injuring agent and allow repair to occur, which is healing. But as we've discussed, it's a potentially harmful process where the components of the inflammation that kill microbes can also actually injure normal tissues in this process as well. So, acute inflammation is the immediate tissue reaction that's characterized by the accumulation of fluid, right? So, immediately fluids start coming to that area. With the fluids come plasma proteins, and also with the fluid comes innate immune cells. And how intense this response is will depend upon the stimulus. It'll depend upon the duration of the stimulus. It'll depend on the genetics of the host factors, like the genetic predisposition of the host, and also medical interventions like has the patient taken any drugs, etc. The causes typically of inflammation include infections. These can be bacterial, viral, fungal, or parasitic, and these can be mild to fatal. And the tissue necrosis that occurs with it is because of ischemia, and it can also be because of thermal injury. Again, foreign bodies can cause inflammation, and this can include splinters, dirt, and sutures. Immune reactions, like a hypersensitivity reaction, can cause inflammation. And all of the cells that are involved in inflammation are given in this fake blood vessel drawing here. There's a neutrophil, a lymphocyte, a monocyte, a neutrophil here again, a basophil, and an eosinophil. So in this blood vessel, you can see that there are endothelial cells on the sides. And in the endothelial cells, you'll develop a gap in the endothelial permeability, and then there will be leakage of these cells. And outside the vessels are macrophages and mast cells. So here is the normal epithelial endothelial barrier, and you can see that the microbes are coming in. Now, when the microbes come in, or if there's necrosis or tissue damage, all of these things can activate by recognition of the macrophages and other sentinel cells in the tissues. So the mast cells, the dendritic cells, and the macrophages can either pick up on the microbes or the tissue damage. So those are the two ways we'll be studying inflammation. So when this happens, these activated cells will release mediators. You can see these cells releasing mediators, which can be amines and cytokines, and they will go to the local blood vessel and they will cause recruitment of leukocytes. And so monocytes will come out and granulocytes will come out. Plasma proteins will, like I discussed, will come out, and also the edema will happen because fluid will come out as well. As a result of this inflammation, either due to the microbes or due to the necrotic tissue, you will have edema, you'll have mediators of inflammation, and you'll have elimination of microbes in dead tissue. So, how do we recognize that there is inflammation that is needed? And they're recognized by the innate immune system. So, the two things that are recognized are the damage associated molecular patterns, or DAMPs, or the protein associated molecular patterns, or PAMPs. These activate toll-like receptors and other recognition receptors on the monocytes. So, here's bacteria. The bacteria can cause I'm sorry about that. The bacteria can cause the formation of pathogen-associated molecular patterns, or it can cause cell death or damage, and that can cause damage-associated molecular patterns. Ultimately, what happens is that it forms a complex called the inflammasome, which is a multi-protein complex characterized by activation of caspase 1. And ultimately, this leads to cleavage of interleukin 1, and that causes an active form that sets the cytokines in motion, and ultimately, a multi-phase cellular inflammatory response ensues. So here you can see there's a protein activated molecular patterns and the damage activated molecular patterns, but basically all of them result in the caspase 1 being activated and interleukin 1 being going from pro to mature and then being released. So here you can remember that the pathogen associated molecular patterns are identified by the toll-like receptors, and the damage associated molecular patterns are identified by other receptors. But ultimately, you can see that the inflammasome has the activation of caspase 1, which causes interleukin 1b to become mature interleukin from a pro form to a mature form. So, what are the recognition factors, the cellular response, and the outcomes? So, here, if you have lipid mediators, chemokines, or N-formal methionine peptides, they'll be recognized by the G protein coupled receptors, and that will cause cytoskeletal changes, and signal transduction will cause adhesion to the endothelium and also migration into tissues. You can have microbes which have lipopolysaccharide or LPS. These will be recognized by the toll-like or the TLR receptors and also with the cytokines. And together they will cause amplification of the inflammatory reaction. And then also bugs can be recognized by the phagocytic receptor. And then that will also cause microbicidal activity of leukocytes and killing of the microbes. So this is what we were discussing, but the first response will be the vascular response, right? The blood vessel will respond. So, what are the critical components of the vascular response? The critical components are that the vasculature is regulated by cytokines, chemokines, and inflammatory mediators. And the idea is that they will expedite innate immune responses to the offending agent. So, the vessels react to the inflammation really quickly so that they can get all of the material there. And one of the ways they do this is by local production of nitric oxide, which is a vasodilator. And this causes dilation of the blood vessels and increases the flow and increased vascular permeability. Plasma proteins like complement and antibodies, they access the extracellular space, and then the cytokines and the chemokines are induced that cause endothelial changes that allow the innate cells to access the inflammatory site. So here in a normal vascular bed, you'll see an arteriole which breaks up into the veins or little venules and small capillaries, and then they're drained by the venule. Now, when there's inflammation, you can see that there's increased blood flow. There's vascular dilation, number one, and that causes increased blood flow. Number two, you have leakage of the plasma proteins that causes edema. And then you have neutrophil emigration, which means that they come out, and that is causes neutrophil accumulation. So going back to this image, here was the little splinter or whatever that causes the bacteria. So the vascular response will be right here in the blood vessel. You'll see that there will be edema, vasodilation, endothelial gaps, and then the cells will come out and then they will destroy the bacteria. So we know that endothelial integrity is very important, and that if you have complement and antibody mediated destruction, even in tissues, even in the cell culture, you can see that gaps start forming in the endothelial layer, and this shows you that antibody and complement can cause gaps in the membrane. So now that the gaps in the membrane have been caused, you can see that in the blood vessel, the neutrophils will start lining up all the way in around in the corners, right? So this is called margination. And the way it happens is that the blood cells are flowing in the middle. And first, they will come towards the side, which is margination. And then, when they start recognizing that they're activated proteins near the site of inflammation, they will start binding with them loosely and then start rolling. And then, once they roll, they'll stop. And then, after stopping, they will go through the membrane and then they'll go to the area of migration. So, this is phase one. And here is a video of this happening. And what I'll do is that I'll try and slow it down just so that you can see it. And so here the white blood cell is in the middle of the field, and then now it goes and slows down and it's now rolling on the endothelium, as you can see, because underneath this endothelium there is inflammation, and all of a sudden you'll see that lots of little proteins come up, and those protein signals will cause adhesion with the white blood cell, and then that will cause, and here you'll see all of them coming out here. This is the marginating neutrophil, and you'll see that ultimately all of these proteins will be activated because this is the vascular response. And as this is coming down, all of those cells will come up, and the cells will start sticking to the membrane receptors here. So let's just keep watching this for a second. It's really slow. Maybe I can speed it up a little bit. And so here these have come up. You can see all of them, and they're going to stop this neutrophil. And then once it stops, it will go through. Okay, now finally it's stopping, and then it will go through inside. Okay. So the rolling adhesion, the tight binding, diaperesis, and migration, this is the way this happens. And basically, this is because of the activations of E-selectins, ICAM, all of these markers. And ultimately, you can remember that interleukin-1 is an inflammatory cytokine that causes this to occur. And basically, the interleukin-6 and TNF-alpha increase all adhesion molecules. So then the question is, how do the polymorphonuclear cells or the neutrophils move from a blood vessel to the point of the inflammation? And it's because of chemotaxis. Chemotaxis is the unidirectional movement along a chemical gradient. And this is caused typically by bacterial peptides. It can also be caused by complement components like C5a and also by the pro-inflammatory cytokine PRETRAD. The chemoattractants that are in the inflammation, they activate membrane receptors on the innate cells and then activate the cytoskeleton to move towards the stimulus. So here in this image, here in this video, you'll see that the
neutrophils, taken from the blood of a graduate student, are mobile cells that will quickly migrate to sites of injury to help fight infection. They are attracted there by chemical signals that are released by other cells of the immune system or by invading microbes. In this experiment, tiny amounts of chemoattractant are released from a micropipette. When neutrophils sense these compounds, they polarize and move towards the source. When the source of the chemoattractant is moved, the neutrophil immediately sends out a new protrusion, and its cell body reorients towards the new location.
So this is the way chemotaxis can actually work with neutrophils. So the second phase is phase two, in which the neutrophils now are degranulating, and then they're causing the death of the offending agent, and then that causes monocytes to come in. Now, monocytes coming in is the next phase, which is phase two. And then ultimately, when the monocytes come in, the monocytes will come in and they will inhibit more neutrophils from coming in. And then basically, the turn off signal will occur and that will inhibit more neutrophils coming. So ultimately, first neutrophils come, then they degranulate, then they cause monocytes to come, and then they stop the neutrophils from coming. So really, when you look at it, the first thing that happens is edema, which is the accumulation of fluid. The next thing that happens is the accumulation of neutrophils. And the third thing that happens is the accumulation of monocytes. So early on in A, you can see neutrophils, these squiggly little cells, and that is because um, this is early inflammation. And then later in the inflammation will be lymphocytes, which will be chronic. So the white cells during an acute inflammation, there's lots of factors that cause white cells to come there. One is the increased delivery because of the fact that there is lots of fluid coming in. The cytokines are going to help these cells respond to the site damage. And in turn, this upregulates families of receptors that cause innate inflammatory cells to migrate across the endothelial lining by margination, adhesion, and transmigration, like I showed you in the video. And the vast majority of the responding cells here are neutrophils. So neutrophils, also known as polymorphonuclear cells or polys, are the most numerous leukocyte in circulation. They are the signature cell of acute inflammation, which is what this lecture is today. And they're capable of rapid production and release by the bone marrow. Their half-life is around 12 minutes in the blood, 12 hours in the blood, and 1 to 2 minutes at the inflammatory site. They are the rapid responders. The marrow responds by pushing mature and slightly immature but functional neutrophils into the blood. And basically, lots of immature cells can also come out. And if immature nucleated cells are coming out, that's called a left shift, which means that immature cells are occurring. Normally, when we think of the maturation of neutrophilic cells, we think of it from left to right. So if immature cells are coming out, you think of that as a left shift, and it's just the way we describe this. So at the site of injury, neutrophils will cause phagocytosis. They will cause recognition and attachment with the C3b coating. They'll cause engulfment and degranulation, and then will cause killing of the molecules that they're attacking. And at the end, when they're about to die, they will throw out the neutrophil extracellular traps, which is another way to kill bacteria. So here you can see that in a neutrophil, this is the bacteria microbe, and it is going to go inside this little vesicle here. And you can see that the microbe is ingested. And here is a lysozyme, and it has enzymes. It will fuse together, and when it fuses, these enzymes are going to destroy the cell by oxidative killing. So there's oxidative killing, and then that will cause the bug to die. So the oxidative burst is caused by activation of NADPH that generates H2O2, which is hydrogen peroxide, which is bleach. And you can see that this all happens within a vesicle within the cell. At the end, the PMN sacrifices its nucleus by opening it up and throwing a net, which is called the neutrophil extracellular trap. And this net will engulf the bacteria and further kill the bacteria and fungi. The second stage is the monocyte stage. And the monocytes are larger cells. They circulate for around 16 hours. They can have granules, but they typically don't. They usually have a folded nucleus and vacuolated blue-gray cytoplasm. And monocytes are in the blood, and the same cell in the body fluid is called a macrophage, and the same cell in a tissue is known as a histiocyte. So the last phase is phase 4, and this is mediated by TGF beta and interleukin 10. And in this phase, there's repair. So control of acute inflammation is very important. It's highly dependent on these monocytes or macrophages. When the inflammatory stimulus is destroyed and there's no toll-like receptor, there's decrease in pro-inflammatory mediators, and there's increase in repair mediators like TGF beta and interleukin 10. So this anti-inflammatory cytokines are established, and then there's healing and wound repair. But And the outcomes of acute inflammation can be resolution and repair. So ultimately, you can restore normal tissue, or it can become chronic inflammation, which will be the next lecture. If you look at acute inflammation, there's a different ways that it can be seen. One is serous inflammation, and this is when you have a transudate. Fibrinous inflammation, where you have lots of fibrin. A suppurative or an abscess inflammation, where there's lots and lots of inflammatory cells. And an ulcerative, where you have mucosa that is being ulcerated. Remember that normally the hydrostatic pressure from inside the blood vessel and the oncotic pressure from outside keeps the blood vessel, all of the materials inside the vessel. Now, in an exudate, you have increased fluid and protein leakage because of the inflammation, and this is an exudate, which will be full of protein and cells. A transudate is basically because of increased hydrostatic pressure and it's fluid leakage. So it does not have lots of proteins or cells, it just has fluid. So here are the different images of the different types of inflammation: serous, fibrinous, abscess, and ulcer. Serous inflammation, here is a picture of the skin and it's a blister, and you can see that there's blistering inflammation, but there's not that much cells. And so because there are not that much cells, it's basically a serous effusion. In contrast, this is fibrinous pericarditis. This forceps is holding open the pericardium, and you can see the heart, and it looks like they describe this as butter on bread. So it looks like buttered bread, but basically this is all fibrin. Fibrin is a large molecule, and this is fibrinous inflammation. Purulent inflammation is when you have abscess or formation or pus, which is basically lots and lots of neutrophils. And lastly, when you have a hole or an ulceration in the mucosa, like in an ulcer, you can see that there's ulcerative inflammation with lots of necrosis and tissue destruction. That's the end of the first lecture, and the next lecture will be chronic inflammation.